Hi there, folks. Welcome to Cell Communication Part 3. Today, we're going to be talking a little bit about hormonal and neuronal signaling. Not too many details. This is not required of you to know for the AP exam, but I feel that hormonal and neuronal signaling are great illustrative examples for everything we've talked so far about signaling pathways. So let's dive right into it. Hormonal signaling, endocrine signaling. Hormones are generated by cells of our endocrine glands and function throughout our bodies. There are two types of hormones, the hydrophilic type, like insulin, because they're usually proteins or peptides. They're unable to cross the plasma membrane, so they have cell surface receptors. But there's a second type of hormones. Those are the small hydrophobic, the steroid hormones. They can freely diffuse through the membrane and their receptors are inside the cells. Lucky for us, the signaling by steroid hormones is actually much simpler than signaling by peptide hormones. But let's review what insulin does. Now, we've talked about insulin and glucose transport a lot. However, I am showing you here this model so that you can actually put things together, the things that we just learned about signaling pathways and the things that we learned about glucose transport. So when blood levels of glucose rise, the pancreas secretes insulin. Insulin will bind to the insulin receptors on cells. And the insulin receptors look a little bit like the EGF receptor, those receptor tyrosine kinases that I showed you earlier on. There's two of them that will link together upon binding to insulin. And as they come together, they will phosphorylate each other. They'll add phosphate groups. Receptor phosphorylates protein 1 phosphorylates protein 2 phosphorylates protein 3. You can see right here. So IRS1 phosphorylates, PI3 kinase phosphorylates, PTEN phosphorylates this, phosphorylates that. So you will be able to see here in this model, this whole phosphorylation cascades. The final kinase here act, once phosphorylated, will actually elicit a cellular response. So what is the response? Now, we learned already about GLUT4 and glucose transporters, right? Glucose transporters get deposited into the membrane. And this increases the rate of diffusion, facilitated diffusion of glucose. So glucose will enter the cell through the GLUT4 transporter. So one response is to increase the rate of glucose diffusion into the cell by simply depositing more GLUT4 transporters. A second response, if you're a liver cell, what are you going to do with all this extra glucose? Well, you're going to turn it into glycogen. Glycogen, the polymer of glucose. It's going to be used to store this extra glucose. So a second response would be to activate an enzyme, and the enzyme is called glycogen synthase. Glycogen synthase will catalyze the dehydration synthesis of glycogen. So it will link glucose monomers to build the glycogen polymer. And this is how insulin works, my friends. Now let's talk about steroid hormones. Steroid hormones such as testosterone, estrogen, and in this case, glucocorticoid, or commonly known as cortisol, are very simple in the way they function. They can freely diffuse through the cell membrane, so their receptor is inside the cell. And it happens to be that their receptor is actually a transcription factor. That's a protein that can bind to DNA, to the promoters of genes, and turn the genes either on or off. In this case, it's a transcriptional activator, so it turns genes on. So the glucocorticoid receptor, or this transcriptional activator right here shown in purple, is bound to this brick in the absence of a signal. That brick, it's another protein that prevents it from entering the nucleus. However, the moment the signal is received, the moment the steroid hormone enters the cell through simple diffusion and binds to its receptor, the glucocorticoid receptor, that brick goes away. That protein is removed, the barrier is removed, and the two of them, the glucocorticoid receptor and glucocorticoid, will enter the nucleus, will bind to promoter sequences of specific genes and turn those genes on. And that, my friends, is the response of the cell to cortisol. Very simple. Signal enters, binds to receptor, receptor goes to the nucleus and turns genes on. There are no signaling cascades, there is no relay molecules. It is simple and elegant. However, I must warn you, these responses take a long time to happen because changes in gene expression take some time. And we're going to talk more about this a little bit later. But now I want to tell you something else, something interesting about hormones. Hormones can have different effects on different cell types. Now, how do you think that is possible? And the example I'm showing you here is that of adrenaline. Epinephrine is another name for adrenaline, the hormone which triggers the fight or flight response. So when threatened, when under stress, we can either stay and fight or we can run away. It has three different responses shown here. On cells of the liver, epinephrine, adrenaline, will trigger glycogen to be broken down to glucose, and glucose leaves those cells, so it raises blood glucose levels. And blood vessels of the skeletal muscle will now dilate, so they'll become wider, which will increase the blood flow to skeletal muscles. On blood vessels of our digestive system, it'll have the exact opposite effect. Those blood vessels will constrict, restricting blood flow to our digestive system. Three different cell types, three different responses. How is that possible? Well, adrenaline binds to the beta receptor on liver cells and the beta receptor on skeletal muscle cells. Same receptor, however, different relay molecules. The proteins inside those cells are different and they elicit different responses. Now, the receptor that is on the intestinal blood vessels is a different receptor. So it's easy to understand why these blood vessels react differently. They respond differently to the same signaling molecule. Okay. Let's go now to neuronal signaling. Now, neuronal signaling is very different. Neurons are very weird in shape, and we already talked about why they're weird, right? They have a high surface area to volume ratio because you have a very thin and a very long axon. That tail coming out of the cell body is very thin and very long. High surface area to volume ratio means very efficient exchange of materials with the environment. And that exchange, we will see soon, is actually ions. And that exchange of ions really is what conducts electrical signals. And the electrical signals are what really are the basis of neuronal signaling and nerve signaling. And you're going to learn more about this if you are enrolled in either neuroscience or anatomy and physiology. But here we're just going to go over some basics of neuronal signaling so you can see how neurons communicate with each other. So neurons, a crucial component in neurons are another type of receptors, which we talked about in the first video, ligand-gated ion channels. Now, these are channels through which ions can pass either into the cell or out of the cell, which are normally closed. They will open, however, upon a signaling molecule, a ligand binding to them. So once a signal is received, these channels will open and will let ions either flow in or flow out of the cell. So what are those signals? Well, those are signals that are transmitted, if you see right here, from one neuron to the next. They're called neurotransmitters. They're chemical signals. These chemical signals will trigger the opening of ligand-gated ion channels. And this is how signals from one neuron are passed through another neuron. This is great, but how are signals passed from here all the way here and from here all the way here? How do signals travel through that axon of neurons? Well, this is made possible by something called the membrane potential. The membrane potential is really a fundamental property of all cells. And cells, what they do is normally they have excess of negative charge on the inside of the cell and excess of positive charge on the outside of the cell. So there's an uneven distribution of charges. Inside is more negative, outside is more positive. The resulting electrical potential is called the resting membrane potential. Now, nerve and muscle cells and certain other cell types can exhibit electrical excitability. So what is this membrane potential and how is it generated? The membrane potential is extremely important for various processes inside the cell, but we're going to discuss it here in the context of neuronal signaling. The membrane potential is maintained by the sodium-potassium pump, which you're already familiar with. It's a protein that uses the energy of ATP to pump 3 sodiums out of the cell and 2 potassiums in, creating an imbalance in charges. Okay, 1/3 of a cell's energy typically is invested in running this sodium-potassium pump. And if you're a neuron, it is actually 2/3 of all of your energy is invested in running that pump. Okay, so the membrane potential is generated by the sodium-potassium pump. And the membrane potential is important to all animal cells. So what does it mean to have a membrane potential though? So the cytoplasm, the cytosol, and the extracellular fluid have different compositions of both positively and negatively charged ions. And an uneven distribution of charges is what the membrane potential is. However, charges do not like to be unevenly distributed. They tend to flow towards each other, right? So positive goes towards the negative, negative towards positive. So even when separated, they want to flow back. And this is called an electrical potential, right? It's a potential energy, the potential of those charges to flow towards each other. This is also known as voltage. And actually, membrane potential is measured in voltage. So now that we know what a membrane potential is, an unequal distribution of charges and the potential to do something, right? Potential energy, electrical potential. How is this used in neuronal signaling? So here's a picture of a cell, and you see how the inside of the cell is negative, the outside is positive. And we have 3 different types of channels, or actually 2 types of channels. And they are voltage gated channels. Now they're not ligand gated, they're voltage gated. It means they open and close if that voltage, if those charges, those differences in charges changes. So the way it is right now, they're closed. The sodium channels are closed, the potassium channels are closed, and this is the resting state of a membrane. If a signal is received, sodium channels will open. Sodium channels open means sodium flows inside the cell. What's going to happen if sodium flows inside the cell? Inside, remember, it's more negative. If you have now positive ions flowing in, you're going to reverse those charges. Now the inside is going to become more positive and the outside is going to become more negative. The more channels, sodium channels open, the more the membrane potential will change. It will reverse charges. At some point, when almost the entirety of the inside is more positive than the outside, and the outside is more negative, this will trigger the potassium channels to open. Once the potassium channels open, now potassium will leave the cell and you will restore the initial state where inside is more negative because now we're keying out positive charges and outside is more positive. Okay, and this is how it looks like. On the x-axis, you will see time. On the y-axis is the membrane potential in millivolts. The resting state is approximately -60. Something triggers the opening of a sodium channel. That something could be a neurotransmitter from another neuron, so it could be a ligand-gated channel, or it could be something from a sensory neuron, so it could be some kind of sensor, whether it's light, touch, heat, certain chemical. The first sodium channels open, the membrane becomes, on the inside, becomes less negative, more positive, because sodium flows inside the cell. Then more and more sodium channels open. These sodium channels are voltage gated channels. So the change in charge will result in more sodium channels open. The inside the cell becomes less negative, less negative, less negative until it reaches zero. Then it becomes positive. When it reaches a certain point, the sodium channels will close, the potassium channels open. Now potassium flows out of the cell, reversing the charges on both sides of the membrane. This is the falling phase. And actually going even a little bit under what it was supposed to be, and eventually restoring the state that it was before. That brief change in charges in the membrane causes ions or charged particles to flow. That is a form of electricity. This will be propagated along the length of the axon, and this is how nerve signals are transmitted. Okay, and again, the ion channels are crucial here. The ion channels are either ligand gated, so a chemical signal binds and they open. They could also be stress activated. The stress could be heat, it could be touch, it could be pressure, or our sensory neurons have them. It could be light, or they're voltage gated. A change in the voltage and the membrane potential can trigger a channel to open. And all of these signals can cause these channels to open and let ions flow back and forth, or in other words, electricity or electrical signals. Okay, now this is totally optional for the curious of you, but those of you that are already enrolled in neuroscience or anatomy, or actually, you don't need to do this because you're going to learn neuronal signaling in a lot more detail in those two classes. But what I've provided you here with is a video explaining how electrical signals are transmitted, and actually an interactive activity about the electrical activity in neurons. So this is it for us for now, and I will see you next time when we talk about feedback loops. Have a great day.